Okay, so hello everyone and welcome to the Target in STEM webinar series in chemistry. I am Anna Kupani from Imperial College London, here today on behalf of Tigers, and it is my great pleasure to introduce our chair for this session, who is Dr. Binuraj Menon. He is a research fellow and group leader in the Department of Chemistry at the University of Warwick, and he is a chemical biologist with research focus on enzymes and biocatalytic and industrial applications. And I will let him say something more about himself. So without further ado, over to you, Binuraj.
Thank you, Anna, for the introduction. I'm chairing this session, and it's a great pleasure for me to be part of this session. So I'm originally from India. I came to UK in 2006 to do my PhD in University of Manchester, and I did as a research associate and a senior research fellow position at the University of Manchester, and a short position at Max Planck Institute. And in 2017, I have started my own independent research group at the University of Warwick. So before we move on to the session, a bit about the housekeeping today. So you should notice that there is a code of contact, which is also available in Tiger's website. So have a look at it, go through the details. And one of the hosts will post the link of those code of contact in the chat section for you to read. By attending this webinar, you are agreeing to abide the code of conduct we have in place. There are two ways you can communicate during this webinar series. First is the chat box. You can use that to chat to the host and to everyone within this room. You can change the settings in the Dropbox menu and type your comments and questions. But please ensure that the discussions or the chat you are doing within the topics we are discussing. Secondly, there will be a time for question and answer at the end of each talk. If you have any specific questions, just type that in the question and answer box, so that we can pick the questions at the end of the talk and ask to the presenter. We are also live capturing these talks today. So if you would like to turn the captions off, you can do that by clicking the CC button in end of the chat window there. We are also grateful to the transcriber 1-to-1 captions for captioning all these talks for us today. We will also recording the webinar series, and it will be available in the next day or two after a few days in Tiger's YouTube channel. Finally, this is a free event, and if you are in a position to donate, please make this donation to Cowrie Scholarship Foundation. They are aiming to fund 100 disadvantaged Black British students through UK Institute in the next decades. Obviously, it's good to mention Tigers before you making any contribution to them. So before we start, let me introduce the first speaker today, George Vine. He's from the University of Cambridge. George has just finished his second year natural science in chemistry. The title of his talk today is Bromodomain Protein in Histone Modification. George, whenever you are ready, have the mics for you.
Great. Thank you so much, Binuraj. Can I just check that everyone can see my slides all right and can hear me fine?
Yes.
Great. Thank you so much. Well, a big thank you firstly to the Tigers for a really eye-opening webinar series so far. I'm going to introduce you to the wild world of epigenetics through my personal favorite example, which is the bromodomain transcription factors. So let's start by explaining that term bromodomain protein, beginning with the protein part, I suppose. For those of you who've tragically missed out on chemical biology so far, a protein is a polymer of amino acids. These are molecules that have both an amino and a carboxyl group. And cells use proteins for everything from catalysis in the case of enzymes to channels to help molecules cross membranes. The list is endless. And what you're seeing on the left here is the bromodomain structural motif. It's packed full of these alpha helices. It's a secondary structure held together by hydrogen bonds. And these helices pack together to form a binding pocket for an acetyl group. It turns out that that's a particularly useful motif in the transcription factors. Those are proteins which work inside your cell nucleus in the first step of gene expression, which is the cell reading a gene in the DNA and creating an RNA copy. And of course, this wouldn't be a chemical biology presentation if I didn't have an unhelpful name, which is that bromodomains have nothing to do with bromine. Just to clear that up, very sorry to upset the halogen enthusiasts in the audience. So why might acetyl binding be a useful property? It turns out that DNA is not a convenient homogeneous double helix. It's actually wrapped around protein complexes called histones to compact the DNA enough to fit into the cell nucleus. And adding an acetyl group to one of these histone complexes has two effects. Firstly, you've got the electrostatic effect. You've suddenly got these nearby negative charges repelling each other, so that makes the DNA between them, as you can see on the top right, much more accessible. And that means proteins can generate more copies, you can increase the level of gene expression. And the second effect is a complexation effect. These acetyl groups raise the affinity for binding to bromodomain proteins. These can then recruit a host of other transcription factors which can further raise that, that gene expression. And epigenetics is effectively the name we give to this property of changing the accessibility and the shape of your genome without actually changing the code. And these epi markers, like the acetyl group, are heritable. They get passed into the daughter cell after each cell division, which gives them the potential to cause some pretty profound changes. And I think that the starkest natural experiment in this occurred during the tragic Dutch hunger winter. That was when, in 1944, the Nazis blocked trains carrying food from entering the Netherlands. And that had particularly stark effects on the pregnant population, because, of course, the famine changed the balance of stress hormones and affected the nutrient levels in pregnant mothers. And blood samples taken from these newborns had a very different pattern of epigenetic markers compared to their siblings who were born before the famine, for instance. And one of the effects of this was the silencing of a gene called PIM3, which appeared to be systemically linked to high obesity rates in these children of famine-linked pregnancies. In fact, to the extent that by the age of 68, these kids had a 10% higher mortality rate than their siblings did. So epigenetics is this strange middle region where effectively the environment has an effect on your genes. And I think that this has a really important message for people like us, the Tigers, involved in LGBT representation and science communication, because we're used to seeing traits as either being genetic and innate or learned environmental factors, sort of nature versus nurture, if you like. But epigenetics really breaks that boundary down, because it shows that decisions that you make throughout your life regarding diet or smoking or stressful situations, these fundamentally affect the way that your genes behave. And that suggests that this nature versus nurture boundary is a lot fuzzier than we see it. And I think this is where the Tigers really have a role to play, because I'm sure we've all seen versions of sort of simplistic public discourse about LGBT identity, the idea that either it's all just down to the toys you played with when you were 5 years old, or it's the sort of crafty handiwork of an elusive gay gene that no one's ever been able to find. And as science communicators, I hope that a more intense understanding of epigenetics and the way that these factors actually interplay, I hope that that can help to build a richer and a more inclusive conversation about these issues. So now that we know what a bromodomain protein is and how epigenetics can act, a useful thing is examining the methods by which we can control epigenetic properties. So what you're seeing on the left is the mode of action for a molecule called JQ1. That's a competitive inhibitor of a subset of bromodomains called the BET proteins. In this case, it blocks the sites that the acetyl group would normally bind, and that means that this bromodomain protein can no longer bring in the other transcription factors that would help to raise that level of gene expression. And you're seeing the structure on the bottom right. That can be particularly useful in diseases that are related to overproliferation, since bromodomains, in this case, are raising the level of gene expression. So this is a potential target therapy in cancers, most obviously, multiple sclerosis, potentially inflammatory disorders. So we've got a very, very wide range of potential applications. But once we have a molecule like JQ1, we're developing it in the lab. How can we quantify its strength as a candidate molecule? Well, one example is a Bromo scan. It's a commercial assay. And as you can see from the diagram, we have a bead to which you've affixed several histone-like motifs, which might be carrying these acetyl groups. And you compare how much bromodomain protein this bead would collect normally to how much protein it collects when you've got your inhibitor present. Obviously, a very high affinity inhibitor will block up all the sites in the bromodomain, so the bead will collect very little of this protein. So the higher affinity, the less protein you collect. And this is a highly sensitive technique. It can quantify dissociation constants in the realm of sort of 100 picomolar. So this is extremely high throughput and high precision. And another tool in the epigenetic toolbox is the histone peptide microarray. This is how scientists can examine what type of histone motif specifically each bromodomain protein prefers to interact with, because there's a lot of diversity around that central structural motif. And what you're seeing from the top right-hand corner is that this particular bromodomain 4 transcription factor interacts much more strongly when the histone protein H4 carries 4 or 5 acetyl groups compared to when it only carries 2 or 3. And the way that you determine this is through the microarray on the left. You immobilize a series of synthetic histones onto a slide, each carrying a slightly different motif, different permutations that the bromodomain might bind to. You then add your bromodomain of interest, and you can stain it with a fluorescent antibody, so that then, as you can see in the top left-hand corner, just by examining the pattern of fluorescence, you can see where the bromodomain is bound, and therefore which histone motif you need to look for in the genome to see where it interacts. So we've got a variety of exciting tools and no end of useful areas in which bromodomains might act. So my name is George Vines, and I appreciate your time. Thank you very much.
Excellent talk, George. I mean, you kind of linked epigenetics with, societal impact and a lot of other stuff. So we still are waiting for more questions here, but one of the things I want to know is like, what is the clinical application of these kind of techniques? I mean, is there any clinical significance? These kind of chips and stuff you mentioned.
Yeah, well, I think it's easier to see experimental applications than clinical ones because of the quite how drastic the effects of giving someone an over-the-counter pill to just inhibit a protein acting at such a fundamental level. It might be that its most valuable application in a purely medical context is just in terms of identifying target genes that we can then approach with more targeted clinical applications. So it might be in vitro studies seeing, okay, if we affect if we look at the epigenetic markers that are associated with a particular signature from this line of cancer cells, that can then help to show up our targets. But from the literature I've seen so far, we're quite a long way from a targeted enough therapy that this could be clinical in the near future. But of course, I'm speaking to many much better informed scientists than me, and I'd love to to find out more if anyone has seen some exciting near future applications.
Thanks, George. Yeah, there's another question from Rachel. So you talked about examples in pregnant women and gene expression in their children. So there are any example, it can go to many generations.
Wow. Oh, goodness. Well, there was, the only example I've seen was and I'll try and track down the paper where this was when, not in humans, but in mice, people were looking at whether the effect of it, because as I saw here, traumatic experiences are very strongly linked to epigenetic modification. And so I think that there was a possibility that in mice, when a cohort of mice were subject to shocks in the presence of, I think, the smell of some sort of petal, then that they induced a sort of Pavlovian fear response to this smell, and that meant that also in the children, in the F1 generation, there was still a remnant of that response, and I think that there were hints that it might have existed as far as F2, but I haven't seen anything extending beyond that, and particularly because of the fact that the vast majority of markers are wiped in the gametes. So it might be that this is an instance of difference between species, because there are certainly some species which have a higher rate of accidental non-wiping of epi markers. So I suppose that there are probably other species besides humans which are more promising to look for those sorts of long-term marking interactions.
So there's another question from Anson. So yeah, he wants to know whether the pollution, the environmental pollution, will make any changes to these epigenetic patterns or something, fossil fuel burning, and is there any evidence for that, or you come across something like that?
Wow, fascinating question. I haven't read anything remotely relevant to it. That sounds very interesting. I think probably because my talk was specifically about bromodomains, which tend to bind to acetyl groups, and those are not largely the markers that are inherited at the same rate as, for example, DNA methylation, which is a much longer lasting epi marker. Perhaps it's just for that reason that very little of the literature I've seen about bromodomains has related to things like long-term effects of pollution on epigenetics, just because it's a more short-term single generation marker. But I'm going to go away and have a long look for marks on that. Thanks very much for the question.
Yeah, okay, thanks. Yeah, so there are a few more questions probably you can answer, in the chat session. So thanks, George, once again for excellent presentation. So
Thank you.
With a lot of important stuff anyway. Thank you. So move on to the next presenter today, Charlotte Mary Istance. She has completed her MChem at University of Manchester and about to start her PhD PhD degree in applied nuclear chemistry. The title of her talk is Double Dagger, a transition of life and gender. So Charlotte, yeah, it's
hello. Sorry, my presentation has just disappeared. There we go. And how's that? Hopefully you can all see that okay. So thank you, Binuraj, for the introduction. As he said, my name is Charlie. My pronouns are she and her, and I've called my presentation Double Dagger. Most of the research I've done in doing my MChem is not for, can't be released because it's either unpublished or it's currently under company, it's classified by the company I was working for. So instead, I'm going to talk a little bit about myself and how I ended up sitting here having just completed a master's degree. So I've been here before, and a few years ago, I was approached by a friend of mine, and she asked me to tell my story. Now, that story didn't contain any lies, but it did contain some omissions. In the last year, I've gone to war with my mental health, and I've shed many, many tears, and I've learned a lot about myself. So this time I'm going to tell you about what I omitted from my story and how I got to where I am today. Omission number one, I wasn't happy as a child. I mean, don't get me wrong, my parents loved me. We weren't hungry or cold or any of the common answers that might follow the question, why were you unhappy? I was unhappy because I used to wet the bed. Every morning, regular as clockwork, I would wake up and find that I hadn't woken up in the night when I needed to. My parents did their best. I was taken to clinics, given tablets, various alarms to wake me up. And I was assured that this was one of those things that children normally grow out of. But with me, it was just taking longer. My young soul began to take its first impressions, loneliness. I felt totally alone in all of this. They told me it happened to other children, but I didn't know any. And I began to feel real fear inside. What if people found out? Society ridiculed people like me, so I couldn't trust anyone with this information. Omission number two. As a child, I felt something was wrong with me. And it wasn't just the bedwetting. Whenever I looked in the mirror, I didn't look right. Oh, that's right. I wasn't a girl. It took me an awful long time to realize I was transgender, and it wasn't an easy thing to do. I grew up in the 1980s, and that's when trans people were portrayed as big, hairy guys stuffed into charity shop dresses with wigs and bad makeup. I also didn't really look much like a boy. I was often called a girl, and it was a source of ridicule. It hurt me so much to think that being a girl was a bad thing. After all, the media portrayed bedwetting as a bad thing and something that shouldn't happen to normal people. So it followed if I'm being called a girl as a form of offense, then that was also a bad thing. And it wasn't until my 30s that I finally came to realize that I was trans and I could start doing something about it. So bedwetting, I looked like a girl. I didn't really trust anyone. I was alone and afraid. Not really the best start in life. The worst part is there wasn't anyone to blame. It was just circumstance. As I grew up, I was often reminded that if I didn't wet the bed, I would be able to go on summer camps, etc. And on the whole, I don't remember many good relationships with other children. I used to hang around with people who bullied me. And this is because I was desperate for any form of attention to try and quash the sense of loneliness inside me. When I hit 16, I finally stopped wetting the bed and I could start to live my life. What's that old expression? Out to the rotovap and into the condenser, or something like that. My parents split up around about the same time as this, when I was 16, and it was 3 weeks for my GCSEs. And I began to break all over again. See, when my dad left, he took with him his passion for travel and exploration, his curiosity and his skill with science and mathematics, and left me with a very angry mother. And she taught me to hate my dad. I don't think that was her intention, but it was certainly the effect. It had a knock-on effect as well, that all of my dad's side of the family, which was my whole family in England, because my mum's family live in Germany and Poland, started to take a dislike to me. And on reflection, that's hardly surprising. From their perspective, my father left for good reasons, which I couldn't see. I felt like a child when I was told I shouldn't feel anger towards my father. No one seemed interested in asking how I felt or why I felt the way I did. It's taken an awfully long time to fix it, but I'm really glad to say I have a good relationship with my father now. But perhaps if I had a more normal childhood without the bedwetting and as yet unknown gender dysphoria, I might have been able to deal with all of this in a much better way. But as it was, I was even more lonely and afraid and constantly paranoid about what other people thought of me. Between 16 and 19, there were 3 long-term relationships and a whole mess of flings in between them. And if I'm honest with you, the lines where one ended and something else began were often a little blurry. I was prey to bottom feeders. In the long-term relationship, I was subjected to different forms of psychological abuse, made to feel small and weak, constantly afraid of the next cold shoulder treatment coming out of nowhere, or forever wondering what might happen if. And the second of these saw constant cheating on me. And I thought so little of myself, I wasn't strong enough to push back and say, I'm done with you, because A, how could I end up with someone so attractive again? And B, maybe the minuscule possibility that I was wrong. It hurts so much to recount these things because I see how little I thought of myself, but also I see how little I was thought of by those around me. No one was good enough of a friend to step forward and say something. Separate to all of this, I had just failed all of my A-levels, and I had to drop out of chemistry early because it clearly wasn't the subject for me. I took a job selling phones, and that's a job where you need self-confidence to push products that people don't want. I finally ended up as a lowly store manager in one of the most backwater stores, losing most of my money each month, train fares. The interesting thing about any form of psychological abuse is that when you're inside it, you don't see it as abuse because you deserve it. To this day, a small part of me questions whether the behaviors of my abusers was really abuse. Because if your head says it wasn't abuse, then their behavior towards me was normal. Did I deserve or cause those events to happen to me? Let's reflect on something here. It's so easy to think, why didn't she just walk away? Well, it's a fair question. But you have to understand that when you're trapped in a place like this, there is no way out. You have to be strong enough to walk away, to leap into the unknown. But when you're so broken or you think so little of yourself, there's no way you would have the strength to. Something big has to change in order for you to make a move. And for me, it was fearing for my life. I was 29 and in the last abusive relationship, living in this little cottage outside of Loughborough. I was desperate to find any way I could escape. And one night, it all blew up. She let all her anger fly at me, and I was so afraid. I went to bed truly afraid. I made a plan to escape. I knew that she was going to drop me off at the station early for work the next day. This was normal. What was unusual was I had some money in my pocket. I would get a taxi back to the house, grab my stuff, and get my mum to come pick me up. And I'm very glad to say that it worked. It wasn't easy, but I did it. I very quickly moved over to my dad's empty house in Leicester as he was on sabbatical abroad. I met someone at work and we started seeing each other. After a while, we got a place of our own in the Leicester city centre, and we travelled. I travelled lots. I took up fencing, worked for a dance company for a while. I was finally starting to live. And I've just lost my place. In the midst of all of this, I read This Book is Gay by Juno Dawson. And it showed me that transgender people were not freaks, creeps, or pervs, but just normal human beings trying to live their best lives, true to themselves. In just a few pages, my life got turned completely upside down. Was that the answer? My partner was excited by the prospect of my transition, and slowly but surely I explored the idea that I might be trans. That wasn't easy though. When you're raised with prejudices, you have to learn to shake them off, even if they're being directed at yourself. I knew I wasn't the stereotype I'd been raised with, but I didn't know how to move forward from there. I took some counseling, and my counselor wasn't really interested in talking about my gender identity. She was more interested in me lifting the lid on all the pain and loneliness I'd lived with, digging out my demons and facing them. For the first time ever, I'd faced the fact that I'd wet the bed, and I sobbed my eyes out. A few years later, my partner and I got married, and we went traveling around New Zealand for our honeymoon. Going to NZ was definitely a bucket list thing for me. One evening, I had visited a hot spring alongside Lake Tekapo, and I was blessed with the most amazing star field I had ever seen. And there in the middle was the Southern Cross, all of these new stars. And I cried again. Whilst taking in this view, I was all by myself in this warm water, and I realized I could do anything I wanted. And this was, I think, my first step towards going back to school. My wife and I had pretty full lives. I became a fencing coach and later the chair of Leicester Fencing Club. Between that, traveling and the dance company, we were always busy, but something was hanging around my neck. We'd only been back from New Zealand for a couple of weeks, and I was at a manager's meeting, and the boss played Al Pacino's speech from Any Given Sunday, where he talks about fighting inch by inch. It's a great speech, but I watched my colleagues getting impassioned about this, about selling phones. They were willing to make it their life's work, and good for them, but it showed me why I hated it being there so much. I couldn't do that. It was draining on my soul. I would go back to school, and I realized it had to be chemistry. It couldn't have been anything but chemistry. I'd heard about an access course, and I went for an interview at Leicester College, and the admissions tutor accepted me there and then. I resat my maths GCSE 2, wanting to improve on the C I had just scraped as a teen. I wrote 45 and 7 on my wall at home, 45 out of 45 distinctions and 7 an A at the GCSE. I also started working for Lush, and this was the place I started living my life as Charlie. Nobody knew what my dead name was, and I was welcomed with love and respect. I worked harder than I ever worked before, and I was able to secure a place in Manchester, having successfully achieved my 45 and my 7. When we moved north in summer of 2017 to start a new life. Studying chemistry was just mind-blowing, but I also found that I got accepted straight away as a cisgendered woman without question. A few people worked it out eventually, but for the most part, no one seemed to know. But this came with its own problems, because through the fear of what other people might think of me, that came back again. And I was terrified of people finding out. And this hurt things at home. I wanted to hide my past as much as possible, and all our wedding stuff got relegated to the bedroom, and I didn't want to talk about it. Once I started my medical transition, I think my wife realized she could no longer stay married to me. What a lot of people don't realize is how many little things change on HRT. You emote differently, you smell differently, and yes, you even taste different. In the end, we separated, and I moved to live with my friend in the city. I was quite worried at this point because I was halfway through a degree, and what do I do for money? Third year would be fine. I had an industrial placement, but what about after that? I tried not to think about it. And I went on a few Tinder dates, got my own place. And then one evening I met my friend for a drink in the gay village in Manchester. And we found ourselves sharing a table with 3 other people. One particular woman I sat opposite, we started talking and we found we shared an awful lot in common. And we got very, very close. It was the start of the most deep and loving relationship I'd ever been a part of. We found in each other a love that is totally unconditional. We both struggled with difficulties in our lives, and we hold each other up and keep each other strong. Leaning on Tina, I've been able to fight back against my demons, become mentally stronger, beat back the feelings of loneliness and lack of self-worth, and finally say, I am enough. So here I am, 38 years old, master's degree in chemistry. I'm still very broken, but I'm much stronger than I've ever been. I understand that I'm grieving for the loss of my younger life. The freedoms that many young people have born from strength and a carefree attitude were lost to me. Fear of what others would say usually led me to isolate and say no. I'm working to come to peace with that. In the meantime, I have the love and support of an amazing woman and amazing friends. I have a PhD position lined up, and Tina and I are in the early stages of fertility treatment to start a family. That's not too bad for a girl who used to hate herself. Thank you very, very much for listening.
Many thanks, Charlie, for coming and speaking this out very loudly. It's really heartbreaking to hear your story, and I think this will resonate for many people to have that courage to come forward. At the same time, we feel really proud for you. You came all this way. Same time, you explored all your passion in traveling, as well as continuing your studies. So, we are, and you should be extremely proud of yourself. Thanks. Yeah.
Thank you very, very much.
So yeah, let's start with some of the questions. So I just want to know, you are doing your PhD as well as you completed your MChem. So have you got enough support from institution, the educational institution you were part of in this journey, or are they supporting you, or they could do much better?
You'll be pleased to know that I've been at the University of Manchester as well, and yeah, they've been really, really fantastic. Like I said, I was very afraid of being open and speaking up about the fact that I was trans, but there were a few people I shared that fact with in the faculty, and all of them were just wonderful, really supportive. And then, as I said, I kept it quiet from my friends, but after I split up with my ex, I started embracing it more and being more open about it. And everyone just said, okay, cool. Is that all you wanted to tell us? And you were building up to something so big and you just wanted to say you were trans, that's fine. And that was it. So good people.
So what attracted you to the chemistry, or what is exactly, you're looking for?
The fact that my A-level chemistry teacher told me, yeah, this is clearly not the subject for you. That had stuck with me from that day to the day I decided to go back to school. I didn't like it. It's difficult to explain what it was, but it was just something didn't feel right about it. And so, yeah, in the end, when I thought I want to go back to school, like I said, I knew it could only have ever been chemistry, because it just bothered me so much that I wasn't good enough for it.
Yeah, so you'll be going to do the nuclear chemistry, right? So
That's
which
right.
part exactly you are focusing on? I mean, this is still a complicated subject, yeah.
It's so this particular work, it's going to be the testing of the backfill material that's used for the long-term geostorage of intermediate nuclear waste. So all of the legacy stuff from the Cold War and nuclear power. We need to identify what's going to happen to that material in 1,000 years, in 10,000 years. So I'll be artificially aging it and then exposing it to uranium, neptunium, plutonium complexes and seeing just how mobile these aqueous compounds are. Basically, so our great-great-great-grandchildren don't have Chernobyl 2 on their hands.
Yeah. So you talked about a lot of confidentiality in your previous research and result, right? So someone want to know, like, can you talk about a bit more? What restricts these kind of publishing result? Yeah.
So in my third year, I was working on a new analytical tool. I was working for AstraZeneca, and this was just at the start of the COVID pandemic. And I was working on an analytical technique for oligonucleotides using low-res mass spectrometry. But the technique is company confidential, so I'm not allowed to talk about what I did, but it was mass spec. I was really interested in pursuing analytical a little bit more because I didn't find it that exciting at uni, but I knew how important it was. So I wanted to go and work at it for a year instead. And then in my fourth year, I did this new area of chemistry of combining frustrated Lewis pair group 14 complexes with group 15 Zintel clusters and trying to achieve small molecule activation in heteroallenes. Really interesting stuff, but again, massive new area of chemistry that the university currently has embargoed until we have some papers out about it.
You have mentioned that, it's now, you are at your, not very young, right? I mean, and you will be studying with young students in your course. So do you find any barrier, is there any barrier because of age, or how you cope with that?
I'm glad to say no. But when I was chair of the fencing club, it was often remarked by the parents that I got on really well with the young fencers as well. And people would ask me, how is it you do that? And I said, well, because I treat them the same as I treat everyone else. Obviously, with children, there are certain conversations you don't have, but I just treat everyone the same, and that's it.
Thanks, Charlie. I'm sure a lot of people would like to ask about science and life, etc. You are an excellent scientist and a human being, so keep it up. And thanks for coming in today. Thank you. Yeah. So let's move on to the next speaker today. Our next speaker is the keynote speaker, and she from Lara Lalemi. I hope I pronounced the name correctly. Lara is a London-based researcher at the University of Bristol with a passion and interest in environmental sciences. And she has done a first-class MSc during her undergraduate degree, and then currently completing her doctorate degree in chemistry. So she's more interested in gaining more confidence in research, and currently studying aerosol science, and finally able to understand a lot of scientific as well as societal problems we are facing now. Throughout her PhD, Lara has led a group of academics from Keele University to Cambridge University, especially in decolonizing science, and as well as conducting workshop in that area. She has also worked as a consultant in British Antarctica survey, and given a lot of talks in different universities and institutes across UK. Lara is also the CEO of Creative Tuition, and striving to increase accessibility and opportunity for many underrepresented young people from all backgrounds in STEM subjects. The title of her talk is Decolonizing My Mind and My Environment. And happy to welcome Lara for her keynote presentation.
Hello, everyone. It's lovely. I do that presentation, or that introduction was lovely. I'm just going to present now. Give me 2 seconds. Okay. Okay. So I'm actually going to be talking to you about my research, and hopefully showing you why it's so important. Well, it's important to me and how it leads into the decolonizing science. So just to start, why I study what I study, I look at aerosols, and aerosols are the, sorry, hang on. My slides are now gone wrong. Let me try and share that again. Apologies, everyone. It's annoying that the keynote speaker has suddenly gone wrong. There you go. Okay, sweet, sorry. Here we go again. There we go. And there you go. Perfect, right. So yes, I look at aerosols, and aerosols have two main effects on our climate. So they have an indirect and direct effect. The direct effect is where they directly absorb or scatter light radiation back into the atmosphere. And the indirect radiative effect is where they modify the microphysical properties of clouds. So that's why I'm here today. Clouds, these big, lovely, fluffy things in the sky. And since I was a child, I always looked up and wondered why they were there. I wondered why the sky was blue. And actually, through my PhD, I've been able to answer these questions. But also why clouds are really important is because we know, or aerosols are also important, is because we know they have a net cooling effect on our own planet, which helps to offset the heating effect through carbon dioxide. But as you can see here, there's a large uncertainty to how much they cause this radiative loss of light radiation. And so it's really important to study the microphysical properties, so the smaller properties of the cloud, of aerosols, so that we can understand the macrophysical properties. So when from aerosols, that they can be released through plumes from factories, cars. They're in high density in polluted areas compared to non-polluted areas. But as the aerosols grow and water condenses onto them, they encounter this barrier, and this is barrier to growth. And so what it means is that if there is not sufficient energy to go surpass the barrier, then the droplet does no longer grows, it actually evaporates back down and becomes either an aerosol, or it completely evaporates completely. So actually, what I'm looking at now is the presence of surfactants in aerosols. So in a lot of my work, I have levitated aerosol particles using lasers and optical forces, using electrical forces. But here, in study I'm using today, it's really just looking at surface tension, because surface tension is actually a property that, if it is decreased, lowers the barrier. So it decreases that barrier towards growing towards that cloud droplet. And like I said, the clouds are really important in our climate. So surfactants, studies have shown the presence of surfactants can lower the barrier, and that means that you are more likely to go over the barrier and get higher growth of or a greater growth of those aerosol particles into cloud droplets, and then eventually into clouds. Now, understanding surface tension in terms of in these terms again can help inform climate models. So we are able to inform the climate predictions better. So a study has shown that in Sweden, the presence of surfactant has actually lowered surface tensions to around 40. Now, if you compare that to water, which is usually what's assumed in climate models, is a significant reduction. So therefore, it's more likely that we're getting a higher percentage, around 30 to 50% more activated droplets into cloud droplets, and therefore that is going to affect our climate and climate predictions. So what do surfactants actually do? Surfactants interrupt the intermolecular forces at the surface of a droplet. Because of this, they can then affect, because in water, the hydrogen bonds and other interfacial bonds are really strong, they interrupt and disrupt these forces, and so therefore lower the surface tension. And if you think surface tension is just a force between the air and the water surface. So I am looking at surfactant concentration, but I also want to look at the size dependency and the surfactant type. The concentration is important because at a lower concentration, you have, compared to a higher concentration, you have different mechanisms at play, stopping the reduction of the surface tension. In size dependency, again, you get a difference between the diffusion distances between the bulk of the droplet compared to the surface. And with surfactant type, when the surfactant molecules diffuse to the surface, they sit within the subsurface, and within that subsurface they have to rearrange and orientate themselves in the correct orientation so that they can adsorb onto the surface. Now, if they have a longer tail, it is more likely for them to get entangled amongst each other. So it takes longer for adsorption to occur. So therefore it takes longer for that surface tension reduction. So all of these factors are really important to really characterizing surface tension and what surfactants, the effects surfactants have on surface tension. And the reason why we do this in the droplet phase is compared to bulk, because a lot of measurements have been done in the bulk. There's the tessiometer measurements, which are pendant drops, and you can do it in the bulk. But the problem with that is, in droplets, which are more ubiquitous in the atmosphere, you see a higher surface to volume ratio. Therefore, you see different pronounced problems in the droplet compared to the bulk. So it's really important, actually, that we study within the bulk. And so I'm just going to quickly run through the method which we use. It's actually the simplest method I have used throughout my studies, yet it's literally the hardest thing to make work. And I don't understand why, but chemistry is one of those things that even the simplest problem can be made complicated in 2.2 seconds. So what I do is I watch oscillations of droplets going across the camera. And then through these oscillations, I can calculate the frequency and then therefore the surface tension, which is here. So this video plays. So yeah, I thought I'd give you a nice little a video of what's happening. So as you can see, oh, no, there you go. So as you can see, the droplet oscillates, and as it goes down and up and down and up, and it changes aspect ratio. Now, aspect ratio means the height over the width, and so what it eventually does is peter out to 1. And we're looking at these, and through that we get a graph such as this. So against droplet lifetime. And then we have to determine when is the L equals 2 mode. So L equals 2 means when it's oscillating both in the same in the width and the height in the same way. So it's oscillating along both the Y and the X axis, compared to the higher order modes, which you'll see at the start, where they're oscillating in multiple different ways. So from this, we have to determine where this point is. When does it hit the L equals 2? And we determine that as a place where the center displacement, which is what our computer program tells us, we find that through when it goes into a more plateau state. So we're around here, that's where we say the L equals 2 kicks into place. And then from that point, we take in the droplet aspect ratio, everything after that, we do a Fourier transform, a fast Fourier transform. Now, as you can see here, the difference between water and SDS, and SDS is actually the surfactant I'm looking at, and that's because it's a good proxy for surfactants we find in the atmosphere. But if you compare water to SDS, so if there were no processes going on within the droplets, so i.e. diffusion and absorption weren't a problem, then you wouldn't see a shift in the frequency over time, or as you chop the data up. But as you can see here, you do see the shift. So it's detailing to us that SES, there is processes going on that are changing the surface tension over time. So as I said, I looked at surfactants, and I looked at around 3, 6, 10, 50, and 100. And it increases in complexity as you go up, because surfactants do not like to behave. They're like naughty children. You tell them to behave and they don't. They fly off everywhere. They run, you turn your head and they're gone, which is not fun, I can tell you that. But what is fun is getting some results. So the CMC for SDS is around 8.1 millimolar. So we've done 2 concentrations below and 2 concentrations above. And so you got 3 and 6, and you see over time, there isn't much of a shift between within the surface tension. They're all around relatively the same number and over time. So this is kind of showing us again, like maybe diffusion is at play here, because it's less concentrated. It means there's less surfactant molecules near the surface, and which means there's a longer distance for them to diffuse. Whereas if you compare this with, I'm not gonna lie, to me, my beautiful above CMC data, because this was a struggle to get, but I loved it nonetheless. As you can see, there's a massive change over time as you increase in concentration from 10 to 100 millimolar. So as you increase the concentration tenfold, but kind of similar behavior. So over time, as time increases, surface tension increases. And so that's kind of telling us that maybe the longer in which the surface has been made. So that's determined from the pinch-off point, from when the droplet leaves the dispenser. We use a micro dispenser. So from that point, we see that as the time increases, surface tension is lower, and that is maybe because there's more time for absorption of the surfactant molecules onto the surface. But we still need to do and take surfactant bulk data, and we then need to run models about this. So in my future work, what I really want to be looking at now is size dependency and surfactant type, because it's really important. And now the talks today have been fantastic. I've really loved learning from both of you. So thank you so much for those talks. And why I think chemistry is so important to me and environmental chemistry is so important to me is because from a young age, I used to see African young babies on the screen crying, and I wanted to just cure them. I was like, what's wrong? Like, why is it like that? And at first, I originally wanted to be a doctor. My brother's actually a plastic surgeon, and I was like, I want to be like him because he's one of my inspirations. And so I wanted to be a doctor. And then when it came to A-levels, I actually failed a lot of my A-levels. When I say fail, it's a fail in the sense that I didn't get the grades in to go to medicine. So I was like, okay, I'll do chemistry, and then I'll go into medicine afterwards. And then I realized I'd be an awful doctor because I don't like standing up all day, and patience. And so I stuck with chemistry, and I started to believe myself with chemistry. And one of the really interesting points is the fact that I think I've gone into environmental science because understanding the environmental impacts that we're currently causing on global South countries is so important. It's so important for me to understand, or for me to the impacts of which chemistry has on the world. And so I started to think, okay, so how can I change my learning environment? So as you said, I'm from the University of Bristol. And it has been a wonderful journey, but also very tumultuous. So I have, in my second year of uni, I looked around, and for some reason, I realized I was literally the only Black girl in my whole degree, in my whole course, in my whole cohort. And I started to think like, oh, okay, I feel slightly isolated. And then I added the fact that like, oh, I think I'm queer. That's great. At the time I came out as bi, but as many people who are of different sexualities will tell you, that sometimes it is a very fluid thing. So at the time I was bi, I was coming out to my parents. That was a whole issue in itself. And I was thinking, well, why isn't there anyone here for me? Who can I talk to about this? At the very least, we should be talking about our environment and how they are breathing a beautiful and environment conducive to learning beyond the subject, as well as learning about yourself. But it wasn't doing that. And so I set up the Being Me in STEM event, and we wrote a report. And this report detailed all the suggestions to the university that they could make to change in which, the environment for BME people for the better. And actually the Pro-Vice Chancellor, Hugh Brady, came and actually informed us that anything we did do, he would love to implement in his work. And eventually they did. But also, looking back, what this actually did was break down that communication line between lecturers and students. It allowed us to talk about things that were really important to us that we never really had the time to talk about, which was race, which was our experiences, which was life. And so this was the start of something really big. And so this kind of culminated to me thinking, so one of the derivatives of that talk, because I wanted to take the suggestions on for myself, was who we see. So often in science, we see these white male pale faces. Totally love those, totally fine to be there. But we don't really get to see any other faces around chemistry. And if you talk to a young student today and you ask them, what does a scientist look like? They probably will draw someone like Darwin or Einstein. They wouldn't draw someone like Lewis Latimer or Alice Bell. They wouldn't draw someone from ancient Egypt or from Islam or from India, where the zero was made, the concept of zero is made. They wouldn't tell you about these things because they don't know. Anyway, so one of the things that I did within my School of Chemistry was I made sure that they put up a board of inspirational Bristol scientists who happened to be BME, because that's a very important point. If you're going to highlight anyone from an LGBT plus background, working class background, disability background, or BME background, it's important that they know that you're not just representing them because of who they are, but because of what they do, what they add to the world. It's such an important point. And another iteration from the Being BME in STEM event that I did was learning about how Eurocentric our curriculums are. So if you consider, we have 3 main curriculums in our education system. So the first is the explicit curriculum, and this is what we provide students in terms of module frameworks, prescribed readings, and assignments. There's the hidden curriculum. Doesn't like me now? Okay. Yeah, there's a hidden curriculum, which is what students learn through not being told. It's the things that are they learn from the dominant culture within schools, within our society, and this is where they learn kind of gender stereotypes. And then there's a null curriculum, which is completely what we just miss out. And you'd be surprised how much we do miss out in our curriculum, especially within the UK, and how hard it is to build this into our curriculum. So notifying myself of this and understanding, okay, the next iteration of talking about race is not just talking about being Black or being BME, it's about, okay, so what are our contributions and how do we enrich our curriculums by adding these stories. So one thing that I did set up was the Decolonising Science workshops. So these workshops really explore what does decolonising science mean. And I'll explain it in a really short analogy now because I've only got really 2 minutes to go. But what decolonising science really means is, imagine if you on the way to Manchester, we'll say we're on the way to Manchester, we're going to visit Charlie. And we are taking this one route. We know there's this one straight road. We know it will get us there. It may be boring. You may see all the same sites you've seen, but you know it's going to get you there. But halfway in between, someone tells you, okay, you have to divert, and you have to divert along the coast. You have to divert in some way you don't want to do, and you're slightly annoyed, you're angry, but okay, you don't really have much of a choice here. And then you get diverted, and then you realize you start to see all these different castles, or you start to see all these different landscapes that you've never seen before. Maybe you stop off at a restaurant that you've never been and try the best food that you've ever had. You go along a little bit longer, and then you stop along and you get ice cream. Wow, the best chocolate ice cream you've tasted in a while. You've never tasted anything like that. I mean, I don't know if it is like that in Manchester. I've never really driven, but the point is you start to see all these things that you would never have seen if you hadn't have taken that detour. Now, decolonising science is that same concept. It's taking you or enriching your journey. It's allowing you to see more of science and the world of science that is just prescribed to you in your Eurocentric education. So, for example, it's not just highlighting BME people and scientists, it's scientific methodology, it's different oral literatures. That's the most important part of it. So it's innovating the scientific curriculum. So another thing that I because of this, and I'm a very much a talk the talk and walk the walk, a passion that I started, which is a non-for-profit organization, was Creative Tuition Collective. And the reason why I started this was because I realized that this information isn't getting filtered down to young people. And we really don't have the time for this government to change. I want students to learn about trans people's lives, learn about LGBT plus lives, learn about political systems, learn about why science exists and how it exists in the history of science. But I want them to learn about it in a fun way. And I also realize that the access to education is quite stark in this country. So I really want to provide that free tuition to young people as well. Access to education shouldn't be behind the paywall. But what I'm most proud of and what I really want to implement is the mental health support, because we want to run mental health support along with this education, because especially coming out of COVID, there's going to be a need for mental health support, and that's what we want to provide for free. So thank you for listening. And if you'd like to contact me, these are my details. I'll leave that up if you need me to. But one quote from I want to share with you from a person that I greatly admire, and I would implore you to go and look at his speeches because they are amazing and they've inspired me. But if you do or you are thinking of making any change that inspires you or can inspire anyone else, and I do encourage that, is that realizing not everything that is changed, not everything that is faced can be changed, but nothing can be changed until it is faced. So thank you for listening, and I welcome your questions.
Thank you, Lara, for your excellent presentation, both science as well as your activities on decolonizing science. So we have like 5 minutes for the questions and to wrap up. So sorry for that, but we could continue the discussion in the later sessions.
Sorry, I thought I'd get all the
No, that's okay. I mean, yeah, it's okay. Thank you. Yeah, so we have quite a lot of questions. First about science. So you think these kind of surfactants can leak to that atmosphere as a pollutant, and play some role in this kind of cloud formation or not?
Really,
yeah.
Surfactants are in our atmosphere regardless, so they're not pollutants. They can be made, so they can be organic surfactants, they're inorganic surfactants. Yeah, so they can be produced through naturally through sea spray, or unnaturally through the anthropogenic releasing of them into the atmosphere. So they are ubiquitous in nature in the atmosphere. It's just the fact that we never factored them in to our climate models until kind of fairly recently.
So your work has any implication for geoengineering or marine sky brightening, etc.? I mean, someone would like to know that.
I get this question all the time. I love it. Geoengineering. Can we put clouds in the atmosphere and hope and pray that light radiation is absorbed? I would love to, but the problem is humans love to change stuff and then they don't realize there's another effect down the line that could be a lot worse. It's actually better that we mitigate our own practices and reduce our anthropogenic activities that are releasing harmful gases into the atmosphere than it is to try to put more clouds into the atmosphere and hope for less, because it affects the hydrological system, and then that affects the oceans and affects ponds, and it affects so many things that just geoengineering is really not enough. It has to really be considered, and I don't think we're at that stage yet.
So you talked about decolonizing curriculum and a lot of stuff you are doing there. So are you getting enough support from the academics or seniors within your department or University of Bristol?
Okay, so it's an interesting question. The University of Bristol have actually set up their own decolonized UAB sector, and they have tried to make courses about it, and they are really being proactive about it. I think I actively chose not to be a part of that because I had already started my own, and I had already been beating against the drum, being like, this needs to be implemented or talked about. And I was often met with, but what does it mean? How do we support it? And but why? And so I was like, for me, I'm a very much person like, I'll just do it myself. So that's what I did. I set up a group, and luckily I was helped actually by a really, really cool academic called Dr. Chris Adams, who gave me the first opportunity to work with these academics, and they're fantastic. They're from like Wolverhampton, Kingston University. And actually what you'll find is in a lot of non-Russell Group universities, they have better practices and are more likely to change and decolonize their curriculum than Russell Group universities. Russell Group universities have somewhat dragged their feet on this subject, and it's because changing the curriculum is quite large, but it's necessary. So yes, I have received support from individuals, but yes, I would love more support. I mean, who wouldn't?
So maybe one more last quick question. So your research has any significance to this transmission of virus, especially the COVID? Yeah. You found any significance or relevance?
Yeah, it does actually. Brilliant question. So actually some people within my group are studying COVID. So they're levitating COVID-filled particles and testing it in different environments, understanding how it works, how it sediments out, temperature effects. But I'm not going to be a spoiler alert. I'm not going to reveal the results because their papers are coming out. But it's actually looking very promising in understanding, getting a complete understanding of what COVID and how COVID interacts in the atmosphere, or how it would in the different environments. And it's actually super interesting to learn about it from a purely scientific point of view. And it's something, yeah, they've definitely been been looking at it. I can't really reveal their data, but always to come, and I'll make sure to share it with Tiger if anyone's interested.
Thank you, Lara. Thanks for your presentation and for your time here. Yeah. So we have to conclude now. So before we conclude, few more information. So our next webinar series is in 2 weeks' time, and it features talk from Haldrin as well as talk from Eleanor Tether. The next keynote speaker in that presentation will be Dr. Jerly Joseph, and the topic will be how cells from compartments without walls. Cells form compartment without walls. So you can sign up for that webinar through Eventbrite. The link for that will be in the chat session in a moment. In the meantime, we would be very grateful for if you could spend a couple of minutes to fill out a short survey and feedback form. One of the hosts will be posting that link as well as the other information about the next webinar in the chat window. Your feedback will be extremely important to evaluate the events as well as conducting future events. So please feel free to fill those forms so that we can follow it up. Thank you to everyone, and thanks for coming here and joining with us today. Thank you.